Assalamualaikum and hello guys, my name is Miss Nadira and in this video I would like to share with you guys the steps involved in gene cloning. The learning outcome for this video is to describe the steps involved in gene cloning by using plasmid. Alright, before we begin, let's look at first what is gene cloning. Basically, gene cloning is a common method used by researchers in molecular biology labs to create multiple copies of a particular gene which contains our gene of interest. Let's recall the basic tools involved in gene cloning. The first one is target DNA with the gene of interest. Second, resurrection enzyme. Next, DNA cloning vector, for example, plasmid. The next is modifying enzyme, for example, DNA ligase, and the last one is host cell, which is the commonly used is E. coli. There are several basic steps involved in gene cloning, started with isolation, cut or cleave, insertion, transformation, amplification, and screening. Let's make it simpler. I-cheetahs. I-C-I-T-A-S. I-cheetahs. The first step is isolation. There are two DNA that we are going to isolate. DNA plasmid from bacterial cell, which is commonly E. coli. Plasmid is a small circular DNA in bacteria that can replicate separately. Another DNA is the target DNA, which contains our gene of interest. It can be taken up from any living cells, for example, animal cells or plant cells. After we isolate the DNA plasmid and the target DNA, now, the both of them will be cut with the same and compatible restriction enzyme. For example, if we use restriction enzyme EcoR1 to cut open the plasmid, we will also use EcoR1 to cut the target DNA into multiple fragments. And remember, one of the DNA fragments contains our gene of interest. So, as the result, we will get one molecule of open plasmid and many molecules of DNA fragments. The gene of interest and the plasmid are now having complementary ends, which is sticky end, or blunt end since they are cut by using the same restriction enzyme. In this diagram, the complementary end is sticky end. Restriction enzyme is like scissors. It cuts at the precise point within recognition site. The example here is EcoR1, which recognizes the recognition site GAATTC and cuts at the precise point which is between the G and the A. As the result, the complementary end is sticky end, or also known as cohesive end. The next step is insertion. The DNA fragment is inserted into the plasmid. Both the target gene and the plasmid are joined by using DNA ligase, which is like a glue that binds these two fragments together by forming phosphodiester bond. So, as the result, we will get recombinant plasmid and non-recombinant plasmids. The next step is transformation. This is done by introducing or returning the plasmids into the host cells. And the common host cells of the bacteria used is E. coli. They are treated with calcium ions and a brief heat shock to create holes in the cell wall, thus making the host cells to become permeable to the plasmid. As the result, there are three possibilities of the host cells. The first one is the host cell that took up recombinant plasmid. It could be with or without the gene of interest. The second one is the host cell with non-recombinant plasmid. And the last one is the host cell without plasmid. Now, all the host cells which are bacteria will be cultured on agar medium. The agar medium is supplemented with ampicillin, which is an antibiotic, and also with X-Cal, which is a molecule that resembling lactose. This technique is called amplification to make multiple copies of our targeted gene, and then the agar plate will be incubated until colonies grow. Please take note, only cells that took up plasmid will reproduce and form colonies. The blue colonies appear because the host cell took up the non-recombinant plasmid. The LECT gene within the plasmid is functional and encodes beta-galactosidase, thus can hydrolyze X-gal. So the blue color appears. The white colonies appear because the host cells took up recombinant plasmid. The LECT gene is disrupted by the insertion of DNA fragment. Thus, the LECT gene is not functioning and cannot hydrolyze X-gal. Once the gene is identified, the clones can be used in many areas of biomedical and industrial research. For example, the use of gene for pest resistance inserted into plants to produce pest-resistant crops. Others, gene is also used to alter bacteria for cleaning up toxic waste. Besides that, protein expressed from the gene of interest can be used for basic research on protein. For example, human growth hormone can be used to treat stunted growth.